International Biology of Conference, Biology ITS. I mean Department of Biology ITS. And at this occasion, we will have a session for invited speaker. And our invited speaker today is Dr. Renat Maya Sofitri. Before having the speech, I read the CV of the invited speaker. I will share screen. Okay. I will read the CV of Bu Maya. She is a lecturer and a senior lecturer and senior researcher at Biology Department ITS. I mean she complete her bachelor degree from Brawijaya University, Universitas Brawijaya in Pest and Disease Plan. And then Bu Maya complete the degree master from UGM, Universitas Gajah Mada, and then she obtained her doctor degree from Max Planck Institute for Marine Microbiology, Bremen, German. And then she have many research experience in microbiology, in biofertilizer, plastic degradation, marine fungi, and etc. Bu Maya, now Bu Maya is Chief of Agriculture and Biotechnology Research Center in Institut Teknologi Sepuluh Nopember, Surabaya. Welcome Bu Maya. Thank you for coming in this session. Hello, Bu Maya.
Yeah, hello. Thank you so much for the invitation.
Okay, and Bu Maya, you have 20 minutes for your speech and 10 minutes for discussion with the participant. Bu Maya, time is yours for delivering your speech.
Okay, thank you so much Bu Wirda and also Bu Dini. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, have a nice good day today. Since this group, this is really mostly or 100% occupied by women, meaning that this group women powerful chat room, yeah? Or not chat room, Zoom room. Okay, just I need a little bit time to do a share screen. Please, just let me use the time for opening my PPT. Okay, still working? Okay. Did you see already my PPT?
Not yet.
Can you see my PPT already?
Not yet.
Not yet?
Oh, wait. Okay.
Okay, good.
Okay, good.
And can you, okay, all of you can read my voice clearly? Okay, good. Thank you, Budini, for your sign. Okay. Okay, now it's still opening. Takes time again. Okay, ladies, I didn't mention the gentlemen here because we are now ladies here. For today, I would like to represent, or I would like to share a little bit about the work that we have been already performing since 4 years ago, and until now we are still working with this one. And I give the title of this talk, Green Management for Agri-Soil Fertility. I'm from the Biology Department of the ITS, and also as mentioned by Bu Wirda already, I'm now working actively under the One Research Center Agri-Food at the ITS. Agri-Food stands for the Agri-Food and Biotechnology. Okay, what is it exactly? This is goes to the agriculture is still one of the most important economic sectors in Indonesia. Please Like Bu Susi already explained a little bit this morning, she suggests us to go to, okay, mari ke laut. Please go to marine, and but now not go to the farming area, but at least now I just talk a little bit about the farming area, because we just realize that agriculture is still the most important economic sector in Indonesia. And also marine side, even they already occupy 75% of the area sectors of the NKRI, seems that the aquaculture is not really well a good established compared with the agriculture. Hoping in the future we can use also, we can improve the aquaculture for the best economic sector for Indonesia. Okay, the agriculture. If you say, if we mention about the agriculture, we say we really know about the extensification and intensification effort in order to increase the production of our agriculture. But we know also the impact of both efforts. For example, for a long-term intensification, we adding a lot of numbers of, for example, artificial or chemical fertilizer, it causing a decrease of the soil fertility, and vice versa also for the extensification. Sometime before we start with this effort, we face already with the difficulty, we found already the land is not fertile. The land is not good for the planting. Now, we seems that this both extensification and intensification will face the vice versa problems. That's why introducing the soil microbes in order to increase soil fertility is really well known. It's really well known as the eco-friendly problem solving. And I would like to line these soil microbes in order to increase soil fertility as my topic for the green management in order increasing soil plant fertility. Okay, this is the macronutrient that we need that needed by the plant. You know, we know already about the NPK factors. This is really important for the soil fertility. N stand for the nitrogen. This is really play important role for the protein and also for the growth of the plant. And the potassium, phosphate stand for the P, phosphate. This atom or this nutrient is really important also for the photosynthesis and also energy conservation. And the potassium itself is really important for enzyme activity. This is one key of the role. And we know already, as already previously mentioned, all these NPK factors or components can be naturally provided by the microbes. That's why the study purpose in our research group, first we would like to isolate and to detect and characterize the microbes. In this case is fungi and bacteria. And also the second, we would like also to apply them, whether they really have the impact to increase the fertility of the soil. Okay, now we go to the first purpose. We went to the one small island, Poteran Island. It is located in Madura Island, and as information, Poteran Island is really occupied mostly by marginal area that really need to be enriched. And from this area, we took samples from the soil samples there, and we isolate the fungi. Not only the fungi, we isolate also the bacteria. And for this area, we get the fungi who can solubilize the phosphate. And the fungi, so you can see from the picture, really beautiful. Mostly they goes to the aspergillus. And we would like also to check whether this fungi has the ability to solubilize the phosphate. We do the assay. This is really a simple assay using the Picoskaya medium. And we see that mostly all the fungi give the index P solubilization after 3 and after the 3 and 4 days incubation. For example, the index really high. They goes to the 6 until the 15, the index. What does it mean? It means that they can use the phosphate, which is really available in the surrounding medium, and the used phosphate will be degraded into soluble phosphate, and the surrounding colony will going to be a clear zone. This is the indication that the text phosphate can be solubilized by the fungi by specific enzyme. Okay, and after that, after doing this one, after doing the simple testing using Picos-Kaya medium, we test again whether this fungi can also live or growth directly until 7 days in the culture using Picos-Kaya as the medium as well. And we see they are growing so well as from the graphics. And after 7 days, you can see they can also solubilize the phosphate until 10 ppm. It is really high numbers. And what does it mean? Meaning that the fungi is really able to provide phosphate for the soil. And you have to remember the soil is coming from the marginal area. Okay. And what is the next? The next also we are working with the bacteria, and from the same sample, the soil sample from the mangrove, and not the mangrove, from the Poteran Island, we got numerous numbers of bacteria. And you can see from the pictures, you can see a lot of numbers, the bacteria growing in our medium. They put the different colors, they put the different size, and also the different margin of the colonies, meaning that even the area is really marginal, I mean, they are still rich with the microbes, especially with the bacteria. And we do again for the bacteria, we would like to know what are they exactly. We are doing the characterization using the biochemical characterization following the Burgess manual for the bacteriology characterization. And they goes to the Bacillus group, they goes to the Cyanobacteria groups, and they goes also to the Staphylococcus group, Enterobacteriaceae, Pseudomonas, and Aeromonas. What does it mean? We got a lot of numbers of genus of the bacteria there. And the similar like fungi that also we test their ability to solubilize the phosphate. Since you can see from the number of the index solubilization here, compared with the fungi, the bacteria give the higher number for solubilizing the phosphate, more strong. The power is more strong. You can see the highest number is 76 performed by the Staphylococcus. And how we do this one? This is again the similar method, simple method. We put on the Picos-Kaya medium and we see the clear zone surrounding the growing colony. Okay, and this is for the comparison that have been already published for one papers dealing with the phosphate solubilizing bacteria and their role in the plant growth promotion. We would like to show you that the isolate bacteria that we have, the power of solubilization is mostly similar with the published method, the published bacteria from the paper. You can see here, they go to the 52, 35, 82, 45, 54, 11, and 16. Even they also reach until 100 numbers, but at least you can see using the calcium phosphate, this bacteria able to solubilize the phosphate compared with the published paper with our marginal area. This is a good sign for me. That's why we are really excited to improve this research. Okay, and then we would like also to know how is the bacteria is really able to degrade the organic compound, for example. And again, we do also the simple testing for them, and we do using the specific medium, we test them whether they can produce the amylolytic, proteolytic, cellulytic, and lipolytic enzyme in order to degrade the glucose for their energy sources. And as you see from the clear zone here, they are really producing the specific enzyme for them and provided the energy from specific organic compound. And also for sure, they will also secrete out any kind of organic compound, which is also really important for the plant growth. This is the graphics about their power to produce the enzyme, the specific enzyme. The index goes from the range to the 4 index. And you can see here the isolate of the bacteria, and all bacteria mostly have minimum 2 enzyme. Especially, mostly they produce much more the proteolytic enzyme because the rice one. Okay. And as I mentioned before then, I just wondering about, okay, from the marginal area, I got a lot of numbers of bacteria and a lot of numbers of fungi who has also the ability to solubilize the phosphate. And we are just thinking about how about I just isolate the rhesus bacteria or bacteria from the fertile or from the planting area, which is really fertile, really rich of the nutrient there. And we do this one, and also we go to the sugar cane plants, to the field, and then we took sample there, we took the soil there, and we got again a lot of numbers of bacteria. And surprisingly, based on all the data here, the data you can hear from the PPT, all the bacteria, for example, we choose now the 6 isolate of them. The 6 isolate of them, they give the index even more higher compared with the marginal area. The phosphate solubilizing, potassium solubilizing, the numbers is up to hundreds. Wow, this is a kind again. Okay, the marginal, we got the bacteria and fungi with the phosphate solubilizing. For the fertility area, we got also the bacteria with the similar power, but with the high number of index. And also in these cases, we try to know whether this all bacteria here mentioned is really able to fix nitrogen. They are not going, they belong to the rhizosphere. We use the fixed nitrogen here. We don't fix the nitrogen as nitrogen, the gas and nitrogen, but we use another nitrogen sources. And we know that they can use this nitrogen sources based on the medium changing color. And also we did the parameters whether this bacteria produce the auxin. And as you see, they are produce the auxin, only 2 of them are not. What does it mean? When I got the bacteria from the fertile one, they are more stronger, the most powerful compared with the marginal one. And also if we do characterization, they goes to the similar genus, Bacillus and Pseudomonas. What does it mean then in our thinking then? That means we can introduce the bacteria from the fertile area to the bacteria to the area which is really marginal, which are not really rich with the nutrient. This is our thinking. Okay, this is okay. I just want to continue about we also do the characterization for the biochemical characters of the Basis manual, and we do a lot numbers of testing. Here is 17, and another one is, okay, I will go further on. Oh, wow, this why so fast. Okay, 17. Okay, no, that's not moving. Okay, until 34 characters of the biochemical characters. And as I mentioned before, based on the biochemical characters, they have been clustered to be a Bacillus and the Pseudomonas, and the three of them goes to Azotobacter. Okay, and this is all the summarize of their ability and also their genus of the 6 of our isolated bacteria from the sugarcane plant. Okay, back again to the second purpose. How about if we apply this bacteria to the real planting and we use the sweet corn planting, and we do a consortium of the most powerful bacteria to be one consortium. We create 3 consortium, the P1, the consortium P1 into the P3, and each consortium consists of different numbers, and maximum numbers is 3 bacteria. And we also want to know whether this is the consortium give the impact, and we create also the control. We create 3 different controls in order to get well known about the impact of the consortium. We do a control T1, we only put the water. A T2, this is totally, we use the chemical fertilizer following the suggested dose. And also the T3, we use only the moles. Okay, how we perform this one? All the consortium, we put it, this is for each consortium. This, we put 50% of the consortium in 1 liter water. This is not really, it is not a sterile water. 50% of consortium, 1% of moles, and 49% of water. We add up, homogenize, and then we put another 4 liter of water, and this become total 5 liter, and that's mean we have the ratio 1 to 50, meaning that the water is dominant of this for this mixture. And from this one, I pour around 100 milli for this planting. This planting has the age of this planting is around 4 weeks. 4 weeks planting the sweet corn 4 weeks seed. Okay, we pour with the 100 milli of this suspension every 3 days for 60 days and every 5 days for 25 days. In between, we still poured with the water only, in order to keep the humidity of the soil. And we take a look. Okay, one thing is I would like to mention here, we use the soil which is not sterile. This is like a soil plant. We can buy the soil plant everywhere. We don't do a sterilization for the soil plant and also for the water. Why we don't do this one? Because we believe that the soil have already indigenous bacteria or indigenous microbes inside, which is also play the important role for the growth of the plant. And we would like to know whether the consortium that have been added also give the positive impact to the indigenous bacteria in order to support the growth of the plant. This is the philosophy behind of this method. Okay, then we go to the plant head, parameter plant head. You can see here. Okay, for the control using the NPK is no doubt at all, because this is really fast to absorb by the plants. The growth at the plant head is really the plant head is really, with the NPK with the control number 2, they are really high enough compared with the others with the another parameter with the another treatments. But here, the treatment of P3, if you see here, P3 is only using consortium. P3 is only using consortium rhizobacteria, consists of 3 isolate without additional NPK at all from the outside, chemical additional NPK. This treatment put the good plant hedge. Because I just wondering if we compare with the control here, control is around here, control here, meaning I would say, I would interpret that this consortium really support the endogenous bacteria to really support, give the positive impact to endogenous bacteria in order to support the growth for the plant heads. This is, okay, control one is only water, meaning that only the endogenous bacteria, they cannot grow without any kind of, even there are bacteria and microbes inside, but if we put the consortium, they grow better compared with the control. Okay, how about the stem diameter of the stem, the primary stem? Again, P3, giving the best hit. Okay, and PK for sure. Okay, and then how about the leaf number and index? Again, P3. And how about again, this is the the leaf, how the white of the leaf, you can see again, P3 have the wider size of the leaf compared with another treatment. Okay, now the weight of the plants. Okay, the weight of the plant, you can see here, this is P3, this is NPK. Again, P3 give the bigger one compared with the NPK control one. And how about the corn cob? This is the most important thing, the production, the corn cob, the production, the crops here. And you can see, this is P3, this is the K2, the NPK, this is only the water, the control, and this one is molasses control. And if we see the length of the crops, again, the P3 is really good. Consists of 3 bacteria, meaning that these 3 bacteria really supporting the endogenous bacteria in order to produce the crops. Okay, the other one is the weight also. You can see from the weight, again, higher for the P3 compared with the control water, but NPK again is the most higher numbers and give the higher impact of the concave. And what is it then? When we open the cups and we see the kernel, this is quite amazing. We see that P1, the kernel, you see, not really good compact. The P2, you see like this. And for the P3, the kernel is really good compared with the K2 using the NPK. What does it mean? Okay, for the control only, the control which is really water, which is only the endogenous bacteria, even they produce the corn cob, but the crop without any kernel. It happened. And also for the K3 here. And we can see here, the kernel really compact compared with this one, but unfortunately only for the size of the corn cob. Quite significantly different between P3 and the NPK. Now, this is really nice having this result. What does it mean then? We would like to say that endogenous bacteria, only endogenous bacteria coming from the fertile area can influence, impact similar way the adding NPK. But the difference is only from the size of the cobs. The kernel similar. Unfortunately, I didn't taste how is the taste of this sweet corn, but my student said the taste was similar between the NPK and P3. Okay, this is quite interesting for us. To continue the work. Okay, I go to the green management for agri-soil fertility to the conclusion. Okay, the conclusion is, are there fungi and bacteria able to provide nitrogen, phosphate, and potassium outside, especially in the marginal area and in the fertile area? Are the bacteria able to degrade complex organic compound into simple organic and inorganic compound? I would say yes, because we proved that one, we got that one. And also, there is the question, another question, are the isolate bacteria supporting the growth of product and productivity of the sweet corn plants? This is the second aim of our work. I would say again, yes. The consortium of the bacteria consists of 3 different bacteria, really put positive impact for the indigenous bacteria supporting the growth of the plants, including the supporting the productivity. But unfortunately, but with the K2 control here, I put also with the K2 control here, still put the best performance. The best performance. That means we are in the, we learn about this one that, okay, why we do this parameter, why we do this treatment, we thought, and we do the treatment only bacteria without adding the NPK, because in our aim, we want to exclude all the NPK from the outside, the NPK, chemical NPK from the outside. But now it's already proven we cannot exclude all the NPK chemical compound from the planting business, because we can see they are growing, but the product is not really not really good enough compared with the chemical one. That's why we are thinking about, okay, now we still have to use the chemical fertilizer, but we just decreasing, lowering the dose of the chemical fertilizer. And this one is again back to, we just try now to promote the possibility using adding basic commercial fertilizer, NPK for example, with a lower dose. Maybe 1/2, 1/4, and 1/5 of the suggested dose. And also we are trying again to go to the improving the application to the field scale, not only in the greenhouse scale using the polybag. We would like to have the field scale. And another things we would like also to try using the marginal soil. What is exactly how the impact our rhizobacteria if we put in the marginal soil, it also influence the similar, even not 100% good, but at least they're good, they give the prospect to improve the fertility of the marginal area, or marginal soil. Okay, this is all from my side. Thank you for listening, hoping that from here arising many discussion to enrich us, especially our research working, to be more applicable. Thank you. I give back to Bu Wirda.
Okay, thank you very much, Bu Maya. We have a nice speech from Bu Maya, and now I'm sure that the audience have many questions, and please write your question in room chat, or please make a question directly if anyone want, please raise your hand. Any question? Any question directly? Or please write your question in room chat. Yes?
Fahru Rozi, I see here your hand.
Yeah,
Okay.
yeah, Bu. Thank you, Prof. Maya, for your nice presentation. I'm Pak Rozi from LIPI. Bu, I just thought a question. So I think it's very good research concerning your fertilizer. But yeah, no, I do also the same way, but in the cocoa fermentation. But do you have some data or method how we can assure that our fertilizer is work on the plant, because in the soil itself they have already indigenous microbe or something like this. And do you have some analysis for that? Okay.
Okay, thank you, Pak Fahru Rozi. I don't understand. I didn't get well the question. Do you mean the analyze? What is it? What do you mean? I analyze the bacteria capability, or what? Or the cocoa plants or something?
No,
Because
for example,
uh-huh.
yeah, for example, in the plant, corn plant, for example, you put the fertilizer with the content of microbe, bacteria, or fungal, something like this. But in the
Yes.
soil itself, they also have indigenous microbe in there. And how we can sure our microbe working on it, or maybe the microbe in the soil is working.
Okay.
Uh,
Yes. Clear enough.
Thank
Thank
you.
you so much for your question. Okay, your question will be addressed by the next research, because
Oh,
for the
okay.
next research we will do like this one. This is really a good question, because we have the similar feeling now, Pak Rozi, because
Okay.
now we divided to the my student doing like this, the one student follow continuing with the proposal, and another student just doing enrichment, uh,
Hmm.
not enrichment, doing the richness and evenness
Okay.
of the indigenous bacteria that have been impacted, that have been influenced by the added rhizobacteria. I could
Okay.
not answer now because I don't have the data, because it will be performed next this semester, because the student is now preparing the research. Hopefully I can give you the data exactly whether the bacteria, the indigenous bacteria, is also becoming the increasing, and the richness and also the evenness is increasing following the adding the rhizobacteria. And another my master students also, she will do the molecular way. I'm still checking the possibility to do this molecular way. For example, doing with the TRFLP,
Okay.
terminal fragment length polymorphisms, in order doing like the fastest way to take a look the effect of the richness and also the evenness of the bacteria before adding and after adding with the several time periods, because we still also believe that the plants also putting something to the rhizosphere area in order to enrich and also to give the food also for the bacteria. Yeah,
Okay.
I'm so sorry, Pak Riza, Pak Fahru Rozi, I could not answer this question at the present time dealing with my research.
Okay.
Okay. Thank you very much, because it's always a question and we are very same interesting. Thank you, Prof. Maya.
Thank
Okay.
you so much.
Okay, thank you, Pak Fahru Rozi. Maybe it's a future research ya, Bu Maya.
Yes.
Okay. Please, your hand. Maybe any other question?
Bu Wida, I'm so sorry, I
We?
can see from the chat, Budini just
We?
chat.
Okay, okay. I will read. Bu Maya, you said that your bacteria have high capacity to solubilizing phosphate because it's come from a marginal land. Does it mean that if we want to isolate a microorganism from nature, we need to make sure that their native environment is very low in nutrient? It's
Okay. Yes, I understood the question. Thank you, Bu
okay.
Wirda. Okay, Budini, maybe this is a little bit confused. Okay, confusing, yeah? Okay, I would like repeat again my explanation. I take two different samples, soil samples. I took the soil sample from the marginal area, which is from the Potran Island, and I took from the really rich, very fertile area, which is for the sugarcane, from the sugarcane planting area. And both area, I got the bacteria who has the ability to do a phosphate solubilization. But the power is different. From the marginal area, from the potteran, the power, the index I got is only maximum 70 index. But from the rich area, from the fertile area, sugarcane area, I got even more than 200 index phosphate solubilization. That means even we isolate from the very margin, not very margin, for the marginal area, we still able to catch bacteria who can solubilize the phosphate. But unfortunately, we don't know about the numbers, the evenness of the bacteria compared with another bacteria who doesn't have this ability. Yeah? Do you understand what I mean? For example, from the margin area, we only got, this is because, please don't forget that we use the laboratorium scale. Sometimes we use a specific medium, then we use the specific medium, and only the bacteria who has this enzyme, they will live in the specific medium, and we decrease, we just moving out the another bacteria. That means we cannot compare how much the evenness between two different areas. Yeah, but I believe from this side, for sure, the evenness of the phosphate solubilizing bacteria from the margin area, maybe for this area, I would say it's really lower number compared to the fertile one.
Okay.
This is answer your question, Bu Adini?
Okay.
Okay, thank you.
Okay. It's clear, Bu Dini? Okay. Okay. Other question? Once more
Hello,
again.
Ibu,
Yes.
can I ask a question?
Okay.
Okay, thank you. I'm Carlia Mehta from SITH ITB. So Ibu Maya, thank you very much for your nice presentation. It was really great to see the results that indigenous bacteria can increase the growth. So maybe it's not really a question. It's just from your presentation, I got interested to see from the plant response, from the side of the plant response. For example, it would be interesting to see how the consortium of P3 bacteria would modulate the expression of genes related to yield, height, and everything. Also, how the antioxidant, the ROS signaling in the roots, for example, as the area that is directly exposed to the bacteria, how is the signaling in the roots change when responding to your consortium of bacteria and to NPK, and how will this in the end cause the good growth of the corn. So yeah, that's not really a question, but then it's just very interesting to see those aspects from the plant sides. Thank you.
Yes, thank you, Bu Carlina. Thank you so much also. That's why the science cannot divide it into, okay, I'm the microbiologist, meaning I don't have anything, any relationship with the botany. No, that's why we always overlap to each other. We are working together. For sure, maybe for the next one, we are dealing with the gene expression for the plant, dealing with any kind of chemical compound from the has already excluded, secreted by the rhizobacteria. But in another aspect for the Wirda and the gangs, the botany labs, but in my side, I would like to do, my students also have the preference doing what kind of the enzyme exactly they secreted to this environment of the rhizobacteria. Not only the enzyme dealing with the phosphate solubilizing, nitrogen fixation, for example, and potassium solubilizing, or the auxin production with another thing, because we believe, we know already, this is that basic theory that bacteria is also releasing the many kind of the small compound, which is also really important for the plants. This is also one of our focus, but for the next. Thank you, Bu Carlina, Bu Carlia.
Okay, thank you, Bu Maya and Bu Carlia. Maybe it's a different focus, Bu Maya, ya, botanis
Yes.
and microbiologist. Okay, we have a very nice presentation and discussion. Thank you, Bu Maya. Thank you
Thank you
all
so much.
participants. But unfortunately, time is limited. So we came in the end of this session. Thank you for your attention, and thank you so much, Bu Maya, for your speech. Thank you.
Thank you also so much. And don't forget, Bu Wirda, can I
Yeah.
add up a little bit? We are really open to discuss via
Ah,
email. You can reach me
wait.
via email. I will write down my email in the chat.
Okay.
My email is Maya, yes?
I can share your email, Bu Maya.
Oh, okay, that's good.
Okay.
This is really good.
If you have a question or join research with Bu Maya, you can contact Bu Maya in maya@bio.its.ic.id or msofitri@gmail.com. Right, Bu Maya? Your
Yes, correct.
email?
Yes,
Okay.
really correct. Thank
Okay.
you, Bu Wirda.
Thank you. You're welcome, Bu Maya. And then I will stop share. Okay. Okay, ladies and gentlemen, we have the other session. The other session. At this occasion, we will have a session for the first parallel session. In this session, have three presenter. Okay. Okay. Maybe I will read the presenter in this parallel session. The first parallel session, I mean, Miss Carlia Meta, and then Miss Junairia, and then Miss Karyanti, Bu Karyanti. Okay, you all in the Zoom room.
Yeah, I'm.
Are you ready in this paper?
Yeah,
In this.
Mbak.
Okay, silakan Bu. Back for the first occasion. No. Before beginning the session, I read the rule. Each presenter have 10 minutes of presentation, followed by 5 minutes of question, answer, or discussion session. Okay. Uh, I will read the question from chat room, and maybe you want to ask or make a question in directly. Please raise your hand. And for the first occasion is for Ibu, I call Ibu ya, Ibu Carlia Metia, with the title Presentation Production of High Antioxidant Microgreen in an Urban Farming System. Okay, for Bu, please, I forget it. Bu Carlia, time is yours for delivering the presentation.
Okay, thank you, Ibu Wirdato. Can I have please access to present my slides?
Okay. Maybe adik-adik mahasiswa can help Bu Carlia.
Okay.
Okay.
So I've been given the okay. So Ibu, is this already the view of my slides on the Zoom?
Yes.
Okay.
It's fine.
So yes, good afternoon, Ibu and Bapak. Thank you for the opportunity from IBOC at ITS for the time for me, Carlia Meta, to present our results on the research of production of high antioxidant microgreens in an urban farming system. We are a team for Sekolah Ilmu dan Teknologi Hayati ITB. So this project was done with our students at the school. So the background of this study is that now we have this very busy life in an urban life, but then most of our activities are sedentary. So especially now we work at home sitting, and then at the break we also sit to watch TV, and then another sit to eat, and etc. So with the high stress and then sedentary lifestyle, not so much of movement, it leads to many lifestyle diseases such as diabetes, which is very high in Indonesia, and then obesity, and then infertility, and so on. And actually, this lifestyle disease is like pressuring our oxidative, our antioxidant system in our body. So it will lead to oxidative stress, that we will need a supply of antioxidants from our food to overcome this problem. And Indonesia, which is an agricultural country, and now urban farming industry is growing in all the big cities, I suppose, in Indonesia. I believe so as well in Surabaya. And then the interest to eat healthy food, so not only for the fullness, but also now consumers is more conscious of the nutrition content of the food, which is now the market for microgreens, which is a small or juvenile, let's say juvenile vegetables, is growing as well in the big cities of Indonesia. Microgreens can be cultivated indoors, so it doesn't need very much big space. Also, we can use just like a shelving in a small room with a natural light, or just a lamp that we use in a room. And its attractiveness relatively higher value than the ordinary vegetable. I mean, not the juvenile ones. So now this urban farming of microgreens has become a big interest in big cities in Indonesia, and with the needs of antioxidants to suppress the oxidative stress. So this means that we are growing microgreens to produce higher antioxidants. For the materials and methods, we will explain a few things, the light combination, and then the assays with the did, also the cultivation, and the statistics analysis. So for the installation, we use like a shelving. So this is a very ordinary shelving that we can get from any hardware store. And then we use a LED lamp installation with a blue and green light. For the treatments, so we use 4 different combinations of the light. The first one is 80% of red light and 13% of blue light. For the second is 80% of red and then 17% of B. And then 43% of red and then 57% of B. And the last one is the 30% of red and then 65% of B. And of course, we also use the white light as the control. So this is the spectrogram of each treatment. As we can see in A and B, it is more, the peak is around 600 nanometer, but then in treatment C and D, the peak is around 400 and so centimeter, which is more in the blue light area. And this is the control, so it's more or less the peak is not in one certain area. As for the cultivation, so we sow the seeds in the medium like this, and then we use a double tray. The first tray, we put some holes in the first tray, and then we put the water on the second tray or on in the bottom tray, and then we put water in the bottom tray, and then we put the second tray with the seeds on the bottom tray like this, and then we put the seeds in the cultivation system of the combination of LED lights. And then after 10 days, we harvest the microgreens of wheat. So here we use wheat, or perhaps more popular as wheatgrass now in healthy food. Okay, as for the assay, we did a total chlorophyll and carotenoid content assay. And then we also did total flavonoid assay, antioxidant activity, so the total antioxidant activity by the ppH assay. And then as for the statistic analysis, we use one-way analysis of variance, followed by Tukey test with a confidence level of a p-value of less than 0.5. And then as for the results, we can see here that, so this is the control, this is A, B, C, and D treatments. Control is the last. We see that actually there is no significant difference between, all these treatments. none of the treatments could induce better weight or better height. Later we see the height, this is the weight. Although C is the, let's say C is the heaviest in the weight, but then it's not really significance. The difference is not significance with control. And actually some combination induce lower weight than the control. And then for the height, it is the same. So no significance difference that we found between all the treatments when compared to control. As for the chlorophyll treatment, here is the control. So no treatments as well could induce higher chlorophyll content, but then some treatments induce lower chlorophyll content. So it's not how we want it. And then for the carotenoid content, the treatment induced higher carotenoid content, which is significant from control, and also significant from other treatments. So we got one positive result for carotenoid content. And then for the flavonoid content, we also got a good results with B treatment, which is around almost more than threefold higher than control, and then significantly higher as well compared to other treatments. And for the antioxidant capacity, also we got a significant higher results with A treatment. So in conclusion, with the growth parameters, it's similar in all treatments. So we didn't get any significant results. And then none of the light combinations significantly improved chlorophyll content when compared to control. As for carotenoid content, increased by light combination of 35 red and 65 blue, flavonoid by 83 red and 17 blue, and total antioxidant capacity by 87% red and 13% blue. So we conclude that each light combination modulated differently the content of chlorophyll, carotenoid, and flavonoid. So we can say one light combination is good for all content of antioxidant. And then unmeasured activity of antioxidants such as catalase or sodium dismutase enzyme and so on could be much higher than the ones tested in the study. And then further development of this cultivation of wheat microgreen could be a promising solution to assist antioxidant supply for urban population, as this is quite simple and doesn't require much space. And it is also considered to be more environmentally because this could reduce the carbon footprints of the farming. So we can do the farming in our home, reducing the carbon during the distribution and processing and so on. So this is our group. And then thank you very much for your attention.
Okay, thank you very much, Bu Carlia. And ladies and gentlemen, the discussion session will be held after the third presenter finish the presentation. And the next presenter is Bu Junairia. Okay, the title is Callus induction of androgropy paniculata with combination of growth regulator NAA and BAP. Oke, Bu Junaria.
Oke, semangat Mas.
Oke, oke. Time is yours for delivering your presentation.
I'm sorry, I cannot share.
Okay.
PPT.
Okay, maybe Aisyah, can you help Bu Junaria?
I'm sorry, we don't have the file. Yes.
Please, Bu Junaria as a co-host. Co-host. Okay. Please, Bu Junaria. Okay.
Yes, please wait.
Okay.
Okay.
Bu Jun, you can start your presentation and your screen share. Okay.
Okay. I'm sorry.
Not at all, Bu Jun. Time is yours.
Okay, ya. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you for this time. I will present careless induction of andrographis paniculata with combination of growth regulators NAA and BAP. The introduction, andrographis paniculata is known to have high medicinal value. This plant contains diterpenoid lactones such as andrographolide. It is used in antiviral, immune enhancement, anti-cancer, anti-inflammatory, analgesic, antioxidant, antimicrobial, hepatoprotective, and cardioprotective. This plant found to contain several important secondary metabolites. Until now, the secondary metabolite is obtained through extraction of organs in plants directly. But this method requires a supply of fresh herbs on a large scale. Furthermore, the process of extraction, isolation, and purification costs are relatively expensive. One of the alternatives that can be done to keep the availability and increasing the production of secondary metabolites in shorter time is through callus culture. The purpose of this research was to know the influence of combination NAA and BAP concentration for inducing of callus from andrographis paniculata leaf explant. This is the figure of andrographis paniculata. Material and methods. The whole plants of andrographis paniculata were collected from the Beratang Flowers Market, Surabaya, East Java, Indonesia. The methods consist of sterilization, culture media, and statistical analysis. Result and discussion consists of callus induction time, callus morphology, and fresh and dry weight of callus. This is table 1, effect of different concentration of NAA and BAP on callus induction, fresh weight and dry weight. This is morphology of andrographis paniculata. Overall, texture of callus is compact. Callus induction time. Callus formation was observed from leaf segment after 6 weeks of inoculation is MS medium supplement with different combination of NAA and BAP. The fastest explant for callus in the MS medium supplement with combination 1 milligram per liter NAA and 2 milligram per liter BAP during 11 days. And the slowest explant for callus formation in the MS medium supplement with combination 1.5 milligram per liter NAA and 2 milligram per liter BAP during 18 days. That higher concentration of cytokinin than auxin on MS medium can induce callus faster because the number of cells undergoing division will increase. The high fresh weight of callus was observed in the MS medium supplement with combination of 2 milligram per liter NAA and 1 milligram per liter BAP. This is fresh weight 0.1920 grams. And the highest direct weight of callus was observed in the MS medium supplement with combination 1.5 milligram per liter NAA and 1.5 milligram per liter BAP with 0.0395 grams. Callus morphology, indicator as plant growth in vitro culture, also can be seen on color and the texture of the callus. Based on the observation suggests that the majority of treatment produce compact callus. Conclusion, based on this research that concentration NAA 1.5 milligram per liter and 1.5 milligram per liter BAP was the best combination for induction of callus from leaf Andrographis paniculata. From me, it's enough. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh. Thank you, Bu Jun. The last occasion, the last presentation in this session is Ibu Karyanti. Ibu Karyanti, are you ready now?
Yeah. I'm ready.
Okay. Time is yours to delivering your presentation.
Okay, I try. Sorry. And my presentation can share. I will share.
Okay.
Yeah. Sorry. Yeah. Thank you for times. I will presentation our research. The title plant micropropagation of potato in liquid media. Our team research in micropropagation laboratory in Biotech Center Serpong, Banten Provinsi. Okay, background our research. The nutrition content of potato is not inferior to rice, so the potato can be used as a source of carbohydrate to replace rice. We know the potato, if we classification, we know two type potatoes. The first, vegetable potatoes. Vegetable potatoes is the potatoes for consumption daily. This potato have variety like granola lembang and granola kembang and others. And two type of potato is the potato industrial. This is potato for food industry. And this is varieties industry potato such Atlantic and Agria and others. Potato production location are scattered in Indonesia. Potato is a source of carbohydrate besides rice. The source of potato seed come from tuber. The demand for potato seed is high. Seed quality degrades with repeat use. Quality and healthy seeds are needed at all times. So tissue culture technology can produce disease-free potato plantlets. In vitro propagation of potato seed generally use solid medium. So liquid medium innovation is designed to reduce production costs in vitro. Purpose our research to get the optimized concentration sucrose in potato propagation in two culture system using liquid media. The first treatment we using two system, aeration and non-aeration, and second treatment using concentration sucrose and the concentration with that sucrose just for control. And the second treatment using sucrose concentration 0.5 and 50 gram per liter. Observation we do every week until the 4th week after planting, and variable observation, high plantlet, number of nodes, number of shoot, and number of root. Methodology our research, the first we prepare preparation culture. The prepare culture we must choose the mother plant. The mother plant we selection. The criteria mother plant is just high productivity and free disease. So next mother plant we bring to laboratory and we induction of buds and installation of bud and do sterilization with chemical natrium hypochlorite, iodine, and alcohol. So next step, we isolation of the apical meristem using a microscope, and the explant we planting in the medium and growing in the grow chamber or grow room, and we have the plantlet and so next step, the plantlet we bring the laboratory analytic to analyza using ELISA to get a plantlet virus free. So next step, we propagation the plantlet free virus in solid medium, and next using source of explant in the treatment. The research observation of shoot length system culture has an effective for shoot length or high shoot, well sucrose concentration a significant effects. The interaction to factors, so the aeration system and all sucrose concentration significant effective for high shoot. The research indicate at the oxygen for the system aeration increase energy intake in spring and increase high shoot. So the effect of the aeration system in a large number of nodes, well, the sucrose concentration obtained within a different number of nodes. Interaction to factor the number of nodes was so that the aeration system treatment at all concentration product more nodes the compare non-aeration. An increase in the number of nodes in influence by aeration, a large number of nodes will produce a large number of seeds. The highest multiplication rate in the aeration system was obtained in the treatment with a sucrose concentration with 7.5 gram per liter. In the compare and non-aeration system with sucrose concentration 15 gram per liter. Indicate the aeration system can reduce the amount of sucrose so as to reduce cost. The use of the aeration system affect the number of shoot with the highest number of shoot in the concentration sucrose 7.5 gram per liter. This research indicate highest number of shoot from the aeration in liquid medium with sucrose concentration 7.5 gram per liter. And is along with the increase the sucrose concentration. So observation of the number of root in the aeration system. So the aeration, the add sucrose increase the number of root, as well aeration system found the aeration system, we can find adventive root. So this is because I think because the plant are under stress condition. So the result visual observation, the all treatment combination in the aeration system with the sucrose, on the shoot morphology, we can now where the shoot look watery, while the other treatment, the shoot morphology normal. For the treatment without aeration, the number shoot in all treatment was less, and the shoot will look short and thin with a lot of adventitious shoot. So the conclusion, our research, the optimal culture system for the production of granular potato plant with morphology, figure, Higgins number, and nodes, and the length of the with aeration, and sucrose the concentration of 705 gram per liter. Thank you for attention.
Okay, thank you, Bu Karyanti. And ladies and gentlemen, we have a nice presentation from Bu Karlia with urban farming, and then Bu Junairia with callus induction of andrographis, and the last from Bu Karyanti with potato, potato for micro propagation. Okay, I'm sure that you all have many questions. Yeah, if you have question, please your hand for question directly or read. Or write in the chat room, please. Any question? Any question directly? Please your hand. Okay, nothing. I will read the question from chat room. Yeah, from Budini again. Thank you, Budini. The question from Budini for Bu Karlia. The question is, it is very interesting research. From the result of your research, do you plan to increase the concentration of antioxidant to be more higher? If so, what kind of treatment do you think can increase the antioxidant in microgreens? Bu Karlia, thank you from Budini. Can you?
Yeah. Yes, sure, Ibu. Thank you,
Okay.
Ibu Dini, for the questions. So yes, the purpose of the research was to induce or to increase the content of antioxidants in the microgreens of wheatgrass. So rumput gandum, perhaps as we say in Bahasa. From our results, the highest increase was recorded when it was treated with the combination of 87% of red light and 13% of blue light with the LED lights. But then for each antioxidant, so and the total antioxidant was that treatment, but then for flavonoid it was from different treatment, and then for carotenoid it was from a different treatment. So what we can conclude here, different combination will induce different antioxidant content differently as well, the modulation also different, but then for total it could be concluded that the best treatment was the 87% of red light and 13% of blue light. So yeah, that would be my answer, Ibu Dini. And somehow, Ibu Dini, sorry, your name seems familiar. Perhaps you were in School of Life Sciences Technology.
Yes.
Some point. Okay.
Maybe you are friends.
I'm not sure. Perhaps, yeah, when I did my master, perhaps Ibu Dini as well.
Yes.
Okay.
Saya menamatkan magister juga di ITB, Bu Karlia.
Iya, Bu. Saya lulus 2010 kalau kita jadi reuni. Maaf.
Okay, the same alumni from ITB.
Yes.
Okay. Thank you, Bu Karlia and Bu Dini. The other question? Is apa ya? Salam hangat from Mr. Triyono. Hello everyone. I want every person here has a very interesting topic and I hope we can collaborate in the near future year based from Mr. Triyono. Thank you Mr. Triyono. Mr. Triyono now in Thailand. Okay. Okay. The other question, please your hand if you ask the make question directly. No, nothing. Okay. Now I have one question from me for Ibu Karyanti. Ibu, how long you can get a plantlet from the explant?
Oh yeah. Thanks to we have the explant sterile, explant free from virus.
Explant meristem, from meristem.
Oh yeah.
Apical,
We done the repair from the induction tuber until we have we just need to 4 until 6 weeks.
Okay. Thank you, Bu Karyanti, because I had tried to make propagation potato, but it's very difficult for me. Okay, thank you. Okay, thank you for your answer. Okay.
Yeah, thank you, Mbak.
The other question? Nothing. To new message again. Okay. Oh no. Any question? Nothing? Okay. Ladies and gentlemen, we have a nice presentation and discussion. Thank you for you. And unfortunately, time is limited. So we come in the end of the session and we will continue the second parallel session. But we have choice. Yeah, we will continue or break before. How about you, Budini? It's continue or break before?
Yeah. Maybe according to the schedule, we will rest for 20 minutes for Isoma.
Okay,
right.
For Isoma. Okay, thank you.
So we will back to the Zoom meeting at jam 1 lebih. Ini sekarang jam 1 kurang 15 ya.
Ya.
Ya. Berarti jam 1 lebih 5 ya.
Okay. Can we take a photo session for?
Okay, photo session.
Yes, for a second. Okay. Please open the camera for a participant. Okay. One, two, three, and wait a second. Sorry. Satu. Okay. Again. 1, 2, 3. Okay. Okay. Thank you for the photo session.
Okay.
Back to Ibu Dini and Ibu Wirda.
Okay. Now we will end the session and see you again in the next parallel session. Thank you.
Thank you, Mbak.
Sami-sami, Bu.